Okay, welcome everyone to cell biology, also known as cytology, the study of cells and the structures within the cells. We have about 50 to 60 trillion cells per human being. The cell is the structural unit of all living things. And when we think about the cell, you can think about 200 different types of cells in the human body. The different types of cells, as you can see on your screen, and if you look in your text, range from a nerve, which helps you to move like so, to skin that covers you, to specialized eye cells. Some cells are worthless, you could say. They just flake off like your skin, but you need them to protect you. And some are vital and just do not regenerate well, like your nerves. If you turn to, in your textbook, to page 73, it's chapter 3, the cell as a city. Many of you have had cell bio in high school, and cell bio includes many different organelles, little structures within a cell. I'm just thinking about what we should do. And I think, what I think I want to do? I hope you don't mind. I think I want to change our plan a little bit because we've lost 20 minutes with computer issues. And it's a big chapter. I think we should do the test on Wednesday. Let me know what you think. I want to change this, and I want to do half the lecture today, half the lecture tomorrow. Cell bio is pretty big, and if we struggle getting through this, it's going to be a struggle. So let's do the test on Thursday at 9. This way we can review a little bit tomorrow and have a little more relaxed of a presentation. All right. I want to just go up to here, the different parts of the cell, and do membrane transport tomorrow. It's a huge chapter. I don't want to overburden you. That's how I am as a teacher. I go with my gut instinct. My gut instinct says we got to hold off and do the test we have plenty of time, and do the test on Thursday. And I will email you your lab for the week, one lab. I want you to concentrate on studying for the test. So that's my decision. If you don't like it or like it, just let me know at the end on that. But I think we'll be better off. Okay, because I'm feeling strained, and that means you're feeling strained. Okay, so let's talk about the different types of cells. I begin the chapter with a story about a girl who has a genetically engineered cell, and makes her better and the other one worse, and she's picked on. So it's a story to get you to read the chapter and think, hey, you can insert different genes into people and have different organelles in people, and it may change our society. That's what it's about. And if you take a look at figure 3.2, it gives a variety of different structures within the cell and how big they are, okay? From the human egg cell to being the largest cell in the human body to tiny atoms. And if you continue to look at the images, I go over a little bit of microscopy, and then we talk about the different types of cells. All right, and let's start with the figure 3.6, the cell as a city. Figure 3.6, and then moving on to figure 3.9. So I think that's the most important figure in the text, 3.9. Basically, the cell theory of life says that all plants and animals are made of cells, and that cells come from pre-existing cells. Cellular, acellular, we call it. Rudolf Virchow came up with that. And my most interesting lab that I ever had was the first lab in cell biology, where we created life. And we put a bunch of phospholipids in a little jar and formed a cell, a little structure similar to a cell. And it's self-assembled, all right? Membranes self-assembled. So we created life. You could say, our first class, we created life. I suppose life as we know it. And phospholipids were what we put together in a jar. Phospholipids have a phosphate head and lipid tails. Phosphate head is what we call hydrophilic or water-loving, and the phosphate tail is hydrophobic or water-fearing. The molecules self-assemble. That means that they form on their own. We believe that's how life formed, that there were precursors to life that developed and formed into cells. All right, now leave it at that, that life could possibly self-assemble. That doesn't mean there's no God. That doesn't mean there is a God. It just means that, whatever created us, however it was created, these phospholipids form a sort of a membrane. And if genetic material gets in, we call them coacervates or primordial pre-existing type cells. But we say that cells have a layer around them of these hydrophobic, hydrophilic phospholipids and cholesterol embedded in between to keep these membranes flexible and moving. This is a mini structure of a membrane. It's made up of about 50% by weight of protein and 50% by weight of phospholipid. Your membrane is your little saran wrap wrapping you, and it's not just like a piece of saran wrap wrapping each and every one of your cells. It is a dynamic saran wrapping. In other words, it lets some things in and other things not. We call it semi-permeable or selectively permeable. It allows some things in, but other things not allowed in. And we say that there are integral proteins, or proteins that are large and in a large mass that carry materials back and forth through the membrane. The anatomy of the integral protein is, as you can see up here, it spans across the entire membrane. And we say that it has hydrophobic and hydrophilic sides. The parts that are on the outside near water are hydrophilic. Let me go back a minute and just re-explain hydrophobic and hydrophilic. To have a phobia means to be afraid of something, right? Philia in Latin means to love it. If you're hydrophilic, that means you're water-loving. In the last class, we said things that are charged are water-loving. So let's go back to our membrane. Our membrane is made up of large channel proteins where the parts that like water are on the outside and the parts that don't like it are on the inside. See these little lipid tails? Lipids are fats. Do they like or hate? Do they like or do they hate water? They hate water. They go away from water. So lipids go to the inside, and to the outside go the hydrophilic or the charged parts. So we say that all membranes have what we call a self-assembled set of phospholipids with embedded membrane proteins in between. These are your phospholipids. These are your membrane proteins. And there are some peripheral proteins. Peripheral means to the outside. Peripheral proteins that kind of mechanically change cells that are not embedded in the membrane. They sit outside as well. And so we call this the fluid mosaic model of structure of a cell. What do we mean by the word fluid? Fluid means moving around. So it's not just the saran wrap that I'm talking about. Things move around and are constantly, dynamically accepting things into a cell and removing things out of a cell. And the proteins act as little ferries or boats in a phospholipid sea. So there's a sea of these little lollipop-looking structures known as phospholipids, like we said, and the proteins bob around in that phospholipid sea to allow things in and out. That is the way in which every cell in our body communicates with other cells. As I've stated before, let's restate. We have about 200 different types of cells in our body. Types. 62 trillion in terms of numbers of those cells. Every one of those cells is a dynamic city, and that is the point of today's lecture, to go over these cells and discuss them as a dynamic city with little parts inside interacting together with a phospholipid membrane. In other words, a fluid mosaic model of a membrane around that allows the cell as a city to communicate with one another and within its own cell. How do we get cells to stick together? Some cells stick together more than others. If you're in the uterus, you better have a pretty tight hold on your cells, right? What comes out of a uterus? A baby's head. So you don't want it flying apart. And if you take a look at desmosomes, that is one type of what we call tight junction, you find them in the heart and in the uterine wall because they are like spot wells. Take a look at the image. They are like welds that hold together two cells to resist tensile forces. And what are the welds made of? They're made up of these anchoring fibers that emanate from the center of the weld. And it's obviously not a real weld. It's obviously a set of proteins that hold cells together really tight. So on the test, I might ask you about cell junctions, and desmosomes are welds that hold cells together really strong. And examples of that are the heart and the uterus, because you want to hold those areas really firm. The heart cannot just fall apart. It has to beat like a trillion times in your life. Tight junctions prevent leaking. Tight junctions are sealants. They have chemicals that seal. In your digestive tract, it's known for having tight junctions. The tight junctions prevent the digestive tract from leaking because your body is really a tube within a tube, isn't it? After all, when you think about your mouth to your anus, it's a tube within a tube. Secondly, I want to add gap junctions. Gap junctions are little hollow tubes that allow connections from one cell to another. Rapid communication. I don't know how you can remember that, except think of, I don't know, the Gap as a store and you're running to the Gap. I don't know. And you will note that gap junctions allow rapid communication in what we call electrically excitable cells. Have you ever had a thought that came to you when you smelled a certain smell? You know, I always think of a certain image of a park when I smell lilacs, or if we just had bacon this morning, I always think of my childhood, a chilly morning, bacon smells good to me, okay? So your mind has a lot of gap junctions. Your brain has gap junctions between your smell and memory that allow instant memory of certain things when you smell certain smells. Those are due to gap junctions, rapid communication between one cell and another. In fact, embryonic cells, embryos are basically your first 8 weeks of life, from when you become a fertilized egg and 8 weeks on. You don't have, at that point in time as an embryo, you don't have a blood vessel system, right? You don't really have much of a nervous system. So gap junctions allow you to communicate from one cell to another when you're an embryo. When we look at cells, we think of cells as having 200 different types. I have a picture of 3 of my favorite. A skin cell here, which is a little flat structure that covers. A nerve cell, which is an electric current. And an intestinal cell, which has a little meat to it. It absorbs and gives off materials. They're 3 cool cells because they're so very different. Different shapes, different sizes, different functions. There's 200 types of those cells. They're all different, but they're all the same. I'm going to highlight how they're all the same. They're all the same in that they all have carbon, oxygen, hydrogen, and nitrogen. 96% of all of your cells are made up of COH and N. They all have the same parts: nucleus, cytoplasm, cell membrane. They are all active cities that are dynamic and perform stable functions to keep homeostasis stable. In other words, to keep homeostasis going, to maintain stability within the body. All do the same general basic thing. And if you can remember anything about this class, remember one thing: disease. Disease equals imbalance of homeostasis, okay? When things go wrong, that's disease. Like the trouble we had with the computer just now, not being able to share the screen. Imbalance of homeostasis. And you see how stressed I get. Now I'm relaxed. But ultimately, that's disease, right? A stressor on the body. If you look at figure 3.9, we've got a picture of all the different, what we call organelles, or structures inside the cell. We call it intracellular structures. The mitochondria, the nucleus, the Golgi apparatus, the lysosome, the vacuole, the ER, known as the endoplasmic reticulum, the ribosomes, all make up organelles within the cell. So our agenda today really is to go over these different organelles, talk about some diseases associated with it, with each of them, and how they function normally in the body. And then in the next class, we'll go over the different types of cell movement, all right? And then we can talk about the test in the next class. And then Thursday, we'll have the test. Okay, so a little change of structure just because we've got to go over these organelles. So why don't we start by making a comparison between the organelles and the different parts of a city. I'm going to say that the mitochondria are your powerhouse of the cell. I'm going to go over each of these, by the way, in some detail. Your mitochondria are your powerhouse of the cell. They make ATP energy. These are little structures, much like in a city having a powerhouse, a coal-fired plant, whatever, solar plant. A mitochondria makes energy. The nucleus is your capital. It stores your DNA. Your DNA, most of us know that, that's your genetic material that controls your cell's functions. Your Golgi apparatus is like your your post office or your church. It packages things and purifies things, you know, gets things ready. Your lysosome is your policeman. It breaks up different groups, cleans things up, you know. You could also call it your garbage man. However you want to say, it's your intracellular digester. It digests materials that need to be removed as waste. And then we have your vacuole, which is your storage units. Storage units can store furniture, right? In a city, stores water, waste, and food in cells. Your endoplasmic reticulum, we call it the ER, okay? And the ER, what it does is it's the subway system of the cell. It transports things moving around. And we say that it looks rough in shape when ribosomes or little protein factories are stuck on the ER. It's called smooth ER when it transports fat and doesn't have that set of ribosomes. Now, I want to say that the cytoplasm of a cell is all your fluid and dissolved materials that organelles are sitting in. So a cell is made up of two things. Cytoplasm, which is also known as your cytosol, all of your fluid and dissolved stuff, and your organelles. So organelles all sit in a liquidy mesh. Each cell has organelles that perform particular tasks, and the cells are compartmentalized by all of these membranes. You have a lot of little compartments working together to carry out all the activities of a cell. I want to begin, now I gave you the overview of all the organelles. I want to begin with each of those organelles and discuss them in detail. As I said, the mitochondria is the power plant of the cell. It makes ATP by cell respiration. This is a picture of the mitochondria. And if we go through, there's another picture in your text, figure 3.13. And if you take a look, the mitochondria has two different membranes, one on the outside, right? And one with folds on the inside. The folds on the inside are known as cristae. And I'm going to tell you a couple of interesting factoids about the mitochondria. It has an inner membrane, the cristae, that looks just like and is structured just like bacteria. It has its own set of ribosomes known as 70S ribosomes, all right? And 70S ribosomes resemble bacterial ribosomes. I'm coming up with the hypothesis here. DNA from the mitochondria resemble DNA from a bacteria. So we say that, and it's called the endosymbiotic theory, we say that mitochondria, we believe, came from bacteria, that we somehow absorbed at some point in the past, we absorbed a bunch of mitochondria, and those mitochondria were primitive bacteria. Figure 3.8 shows the endosymbiotic theory. Figure 3.8 in your text. But I think what you should know as a class, that this is your powerhouse of the cell, and that these mitochondria have their own set of diseases, from chronic sweating to diabetes. We're not sure, but we say that it is for sure self-reproducing. They have their own DNA and RNA to reproduce on their own. So for example, when you're bodybuilding, when you're exercising, you don't build more muscle. You split your mitochondria in half and make your muscle more efficient. So your mitochondria divide in half. And that's the mitochondria's anatomy. ATP produced from cell respiration, the powerhouse of the cell. Most people have memorized that mitochondria are the powerhouse of the cell, but no, most people don't know that your mitochondria reproduce on their own and are their own kind of little self-sustaining unit inside of your body, where you give it a home and it divides on its own. It gives you the energy you need. And if anyone in their class doesn't like their mama, they better think twice, because mitochondria are inherited from your mama, and they're in each of your cells, and they're dividing and giving you all your energy of life. All right? Dad doesn't give you your mitochondria. The sperm from your dad don't have it, that you get it, but the egg from your mom gives you the mitochondria, and it continues to divide to this day. So that's an interesting point. Whenever you have a fight with mama, she can say your mitochondria, or her mitochondria are in you, and that pretty much shuts you down. So, okay, that's kind of tough pill to swallow. All right, ribosomes. Why do I always ask a question on ribosomes on a test? Because ribosomes make protein. Protein do everything in the body. Proteins are basically pumped out by your little dots of ribosomes strewn throughout your body. The ribosomes are made up of RNA, ribonucleic acid, and they are made by structures known as the nucleolus in your nucleus. So proteins do everything in your body. Remember, we talked in the last class about globular and fibrous proteins? Well, enzymes, hair, antibodies, all made of proteins. And what makes proteins? Ribosomes. So I find that that's an important structure to be aware of. Let's talk about the endoplasmic reticulum. As you can see, this is your subway system of a cell. It transports materials through a cell. It provides compartmentalization, in other words, allows a lot of little compartments to form. As you can see, if you put one ER next to another, it allows, it's kind of the biggest word I know, compartmentalization, allows a lot of little compartments to be in your cell, where each little compartment is doing its own thing. And it also provides a surface for enzymes to work on. That surface allows activities to happen. You've got to have surfaces for enzymes to work on, and the ER gives a wet and stable surface in your cells to do certain jobs. We'll leave it at that. The Golgi apparatus purifies, you could say it's the church, purifies materials as they go through, packages them, modifies them, and it moves from a cis to trans side. Looks like a series of flattened pancakes. That's what the Golgi apparatus reminds me of, where I just want to put maple syrup on and eat them up. You know, it's crazy. But anyway, the Golgi apparatus purifies materials. And there's a picture of the ER on figure 3.16, picture of the Golgi on 3.17, and a picture of the mitochondria on 3.13. And a picture on figure 3.11 of the fluid mosaic model. So if you're following along in your text, those are the pictures that I would refer you to. The lysosome is kind of the policeman of a cell because it breaks things down, breaks up crowds. The lysosome is responsible. Its physiology is strictly intracellular digestion. That is, digesting things in your cell that it needs to eat or waste that it needs to remove. We say that the aging process, also known as senescence, is a result of lysosomes that are leaking. Cells of our body, membranes, as you can see, don't stay forever. So a cell just usually doesn't live more than 100 years, Some cells die off really quick. Like red blood cells live about, 3 months, 4 months. But cells just fall apart over time. And we say that the aging process really is the loss of cell function, don't we? I mean, after all, by age 80, you've lost 50% of your kidney's functionality, and that's normal. So aging is the steady loss of cell function. And it is due to literally lysosomes breaking apart, and I hate to say it, digesting yourself from the inside out. You lose function because lysosomes, membranes fall apart, and you start digesting your own cells, and things kind of deteriorate away. That's a way of looking at the aging process. It's, the wear and tear hypothesis of the aging process. Some other things that relate to lysosomes, Tay-Sachs disease is a disorder of the lysosomes, autoimmune disease where the body attacks itself. Wiggle your fingers, and I always during class give a +1 on the test to someone who has an attached finger or toe. When you are in the womb as a fetus, your lysosomes digest the material in between your fingers. So you're born, with individual digits. Otherwise, you'd have webbed toes and feet, all right? Another picture you can look at is figure 3.18. That's an amoeba, but it's similar to an immune cell digesting an enemy like a bacteria. That's how your immune cells work. The lysosomes digest the enemy. Moving along, let's take a look now at the cytoskeleton. Figure 3.13 shows the cytoskeleton and the different fibers in the cytoskeleton. The cytoskeleton is really the cell skeleton. It's the surface on which all organelles sit. In other words, it's got a bunch of rods, 3 types of rods: microtubules, microfilaments, and intermediate fibers. The 3 types of rods that hold together all the organelles. Otherwise, when you make a left turn, all your organelles would just fly to the right side of the cell and you'd fall apart. You need these fibers to hold together the different organelles. Your microtubules are your largest, your microfilaments are your smallest, and your intermediate fibers are in between in size. Each have kind of different functions in the body. The microfilaments are involved in muscle movement, when your muscles contract. Whereas microtubules make up, your hairs in your nose, and the intermediate fibers make up the desmosomes. That's what gives the strength to those desmosomes that I talked about earlier. Again, cilia and flagella, the short hairs of your nose and your respiratory tract, and flagella, which is more or less long whip-like tails, are all made from microtubules. And we say that centrioles, a little structure made of microtubules, gives rise to the cilia and flagella of your body. So you have hairs, and we don't want to forget about the hairs as being an organelle of your body. The most important organelle, one might say, is your nucleus, which has your genetic or hereditary information. It has chromatin. It's thrown about in the form of DNA, and the DNA is your genetic code of life. Most of us know that. That's why I kind of call it the capital of the cell. It's the control center of the cell, because it sends out what we call mRNA, which is messenger RNA, to control the different activities of a cell. Most cells only have one nucleus, one cell, one nucleus, but some have many, like liver cells and skeletal cells. They just do a lot of things. Like the skeletal muscle cells are very big. And red blood cells don't have any nucleus. So there's many reasons for that. We'll talk about that in A&P too. The membrane of the nucleus is double membrane. That is because the capital head better be well protective. It's very sensitive to genetic material. But large nuclear pores that are kind of eight-sided in shape, octagonal pores, will allow materials in and out of a cell. Last but not least, if you look at figure 3.12, there's little organelles known as nucleoluses. Actually, 3.14. Figure 3.14 shows a nucleolus, a little more detailed. A nucleolus is a structure inside of the nucleus, a round structure that makes ribosomes. That's really all you have to know about that. It makes ribosomes. It has loops of DNA that code for ribosomal RNA. You have one or two per nucleus. I wanted to stop here at membrane transport, all right? And we will resume this lecture on membrane transport in the next class. We're about 8 minutes early. I just want to show you what we're going to do in the next class. Passive and active transport. Basically, I want you to read the rest of chapter 3 to look at the different ways in which things move inside of a cell, all right? And this will make it a little more enjoyable the next class, because we're going to go over and talk about cell growth and cancer, and these are not topics to be taken lightly. I want you to understand the characteristics of cancer, and you really need to finish up reading your chapter for that. What I'm going to do now is open up the, I'm going to unmute everyone. Let's see. And then I want to take any chat room questions that you have, and we'll go from there. The test is not tomorrow. Test is, I'm going to write this down, Thursday, 9 a.m., test number 1, emailed to you. And we won't have, what we won't have is a lecture that day. People get totally burned out. As you can see, this is a long lecture today, and I didn't want to, I did not want to overburden you today. All right, I'm going to unmute people. So mute all. Let's see. I just want to make sure I can unmute people. I don't know. The new setup is a little difficult for me because of the fact that I've changed my operating system. So it's like little quirks. So maybe, are you able to unmute yourself? Maybe someone can unmute themselves. Are you able to or no? I don't know. Someone try to unmute yourself. Hopefully, we can't unmute ourselves. All right, see, that's a problem. I gotta, okay, here we go. Okay, I now found the button. You see, it's changed it. Allow to unmute. Okay, sorry about that. Okay, everybody. Now you're all unmuted if you want. Okay, I'm pretty sure. Cameron, why don't you say something?
Hi.
Hi, all right, thank you. Sorry about that. Okay, Tanya has her question. Hey, the test will be on only chapters 11, 3, and what else? Well, chapter 2 you forgot, because that's chemistry. Chapter 3 is today, and chapter 11, those pages. Mainly the notes. Remember, I take things from the notes. So Tanya's got it. And, what I just want to say is when I test, I mostly take from the notes. I made up the test. It should really be 40 minutes, 45 questions. Excuse me, 40 questions, 40. Hold on one moment. One moment. Anyway, the cat is attacking the window, so I have to get him away. He aggravates me every time I lecture. Anyway, the trouble working from home, right? Who knows what you guys have to go through? But anyway, getting back, yes, it'll be 50 minutes for 40 questions. And I was originally thinking 40 questions, 45 minutes, but I'm going to do 50 minutes, 40 questions. 25 questions will be 35 minutes. Everything gets 10 points more than a minute a question. That's how I'm going to do it. 10 minutes more than a minute a question. There should be no rush. I don't want you nervous. A couple of you were a little nervous. I think it's too much time giving a whole hour for that. So anyway, moving on. So I'm going to email you the test 9 o'clock Thursday, and that's, and you'll have 50 minutes to do it, and you'll email me just the answer sheet back. That's how we'll do that. And I usually have the test answered pretty quick. I'm going to email all of you the lab for this week. I don't know, am I wrong? Are you happy with having the test Thursday instead of tomorrow? I mean, we could take the next hour and go through all this, but I don't think it's necessary because we have we have 4 more weeks for getting approximately 5 or 6 lectures done. So we're not in a panic. We've got time. But let me know if you want it now. If everyone agrees, I could do the lecture right now and finish up, but I don't know. If you want to pursue that, we can do it. You said this will end at July 25th, right? Is that a Thursday? July 25th is the last day of class. So we've got 3, 6, 9, 10, 11 more classes left, which is a lot of time to get through 6 lectures. But, we have to give test time too. And I think the last week will be mostly tests. It'll be your practical and your last test. So what else? Sorry about all the disruption. It'll be worked out. Anyway, give it another time as a dry run, and I think we'll be okay. I'll put this up on YouTube, okay, if you want to see it again, and we'll go from there. And I'll email you right now your lab, so you can check that out. That's an introduction. I may as well talk about the lab briefly. It's an introduction to what we're going to do tomorrow. So I think that's not a bad idea. It's showing you demos and going over cell how things move inside of a cell. It's called our Cell Permeability Lab. It talked about basically how things move in a cell. If you put a bunch of stuff in a sack and put it submerged in some water, how do things move in and out of that sack? So the sack acts as a cell. It talks about India ink and Brownian motion, that all motion is vibrating and you just don't see it. And during lab, this is called a wet lab. I usually have a wet lab where you do this. In this case, I decided I could have sent you home a kit so you actually have some of these little bags and you do it. But I'm having you just observe it and follow along in the lab manual, and that's how I'm going to do it now. Next lab I think is neat. That's next week. I'm going to do only 1 lab this week because you have a test, and 1 lab next week because then you'll have a practical. The lab will be on histology, different types of tissues, and then that'll be your midterm practical. And it'll give us time to answer questions. So tomorrow you may have questions. Bring questions if you have any questions on the material we did so far. So this way we don't rush the test tomorrow. I think I'm comfortable with it. I'm not seeing anything on the chat room that you want to not postpone the test. It's not a big postponement. It's just a Thursday test. So that's why we got a fresh start next week. Next week we're going to do histology and and the immune system. And then the following week we'll have a test. So goody gumdrops. Any last questions before we go? All right, I think I'll end the meeting, and I think I know what I'm doing with the new setup for Zoom, okay? So sorry for the delay. All right, look, email me if you have any questions, and we'll go from there.
Thank you.
Okay, bye-bye.